We saw briefly when we looked at plant tissues that an organ is simply a number of tissues that work together to perform a particular function. When we look at plants, there are three major organs: your roots, your stems, and your leaves. The flowers and fruits are also organs, but each of them has evolved from modified leaves or modified stems. So your flowers are actually just modified leaves, and your fruit is a modified stem. The function of each of these plant organs is pretty straightforward. So your leaves are there to photosynthesize. Stems provide support as well as transport of nutrients and water. The roots are responsible for anchoring the plant in the ground so that it doesn't blow away or get washed away. It's also responsible for absorbing water. It is a storage organ, so a lot of the starch that is produced by the plant through photosynthesis is stored. And it's also conduction, so it conducts water in and out. Your flowers obviously are required for reproduction, and fruit are there to ensure that your seeds get dispersed. When we look at the structure of a plant is divided into the shoot and the root. And the shoot is where we find our leaves and stems, and the root obviously is the roots. There are a number of terms which you will find helpful to know. They are not officially in your syllabus, but would be useful when it comes to trying to talk about plant tissues and plant organs. So, for example, we've already spoken about the terminal bud and the axillary bud. The terminal bud being the one at the very top of the shoot, and the axillary bud being the one that gives rise to your lateral branches. When we talk about a leaf, it's useful to know that this portion of the leaf is called the blade, and the little stalk that connects the leaf to the stem is called the petiole. And in fact, when we refer to leaf, what we're referring to is both the petiole and the blade. For interest's sake, this is the midrib of the leaf, and these are your lateral ribs, and those are vascular tissue. When we look at the stem, you will find that the leaves are always present at a point that is called the node. So there is a node from which leaves will grow, and the space in between that is called the internode, between the nodes. And when we look at things like cauliflower, kale, broccoli, etc., all of those plants come from the same plant, which is wild mustard. And all that we've done there is we have selectively bred plants that have, for example, if we're looking at your broccoli, that the internodes are very, very short. For others, we've bred them for having a very long internode. So, for example, if you look at Brussels sprouts come from the same plant that cauliflower and the rest come from, and they have a very long internode. So these are things that are helpful. Knowing these terms, node, internode, petiole, blade, are very useful later on and in matric where we come to talk about evolution. When we're looking at your roots, this is very obviously a dicotyledonous plant because there is a tap root from which we then have lateral roots. These are not adventitious roots. Okay, adventitious roots would come directly out the base of the stem. So this is a tap root or a primary root. All right, let's have a look at each of our three major organs. So when we look at leaves, this term dorsiventral or dorsoventral, either spelling is acceptable. What that means is that the top and the bottom of the leaf are different. So if you have a look at the picture here, you can see that the bottom of the leaf is very pale green, the top of the leaf is very dark green. The rib, the main rib, stands out on the bottom, whereas on the top it kind of folds inwards on itself. So a dorsiventral leaf means that the top and the bottom are different. The leaf is also flattened dorsiventrally, so from top to bottom. It means it's very thin, but has a large surface area. And when we do a cross-section through, you will see that there are actually different regions inside the leaf, which is why the top and the bottom surfaces are different. When we cut through and we do a cross-section of a leaf, this is what we would see. And obviously, we're only looking at dicot leaves. Okay, you do not need to know the monocot leaf. So when we look at the dicot leaf, you'll be very pleased to know that you will not be asked to draw this, but you will be asked to label it. So let's have a look. If we talk about a plan diagram is what you will be asked to draw. It's an outline of what you have observed. It shows the positions of the tissues. It doesn't show individual cells. So that diagram I just showed you now, that is what a plan diagram of it would look like. The only cells that we're drawing are these little guard cells at the bottom because they are very crucial, but the rest of it is simply a region showing different regions. So if we have a look, this here is your vascular bundle, this whole thing. Okay, you can identify the xylem vessels in there, and obviously here you have some phloem. What they're not showing terribly clearly on here are your sclerenchyma, which would obviously sheathe and protect that. These are all parenchyma cells, all of these, but these palisade parenchyma and these spongy mesophyll parenchyma are actually green in color. And that green color means that these, although they are parenchyma, because they have chloroplasts, we call them chlorenchyma. Okay, so we've got your epidermis along the top. Underneath that, your cuticle right on top. Underneath that is your epidermis. They have drawn the epidermis in green, which is incorrect, but I suppose I understand why they're using color. Your epidermis is actually transparent to allow light to come through. These chlorenchyma cells here, your parenchyma, your palisade parenchyma, these are very long rectangular cells. The reason for that is that it actually allows sunlight to penetrate through more easily to get to the depth of or to get to the center part of the leaf. So you've got a region there, a top layer, all of this essentially, which is called palisade parenchyma. Palisade, if you think of palisade fencing, which has those uprights with large gaps between them. That's essentially what this looks like. Underneath that, we have a layer over here, all of this, which is known as your spongy parenchyma or your spongy mesophyll. Notice the large intercellular air spaces between these cells. Notice also that they are more squarish in shape. They're not long and thin, obviously with lots of chloroplasts and that sort of thing as well. The purpose of these intercellular air spaces is for gaseous exchange to take place. So here is a guard cell, there's a guard cell, and in the middle, that space is a stoma. There are no guard cells or stoma, or stomata rather, plural, on that top surface. They're only present on the bottom surface. And the reason is that gaseous exchange is taking place, so gases are moving in and out, which means that the plant is also going to be losing water. And you want to minimize the amount of water that the plant is going to lose through evaporation. So these stomata are only found on the lower surface of your mesophytes. If we're talking about a hydrophyte, a plant that grows in water, obviously that's different, because then this surface would be lying in the water, and so gaseous exchange would not be able to take place. So in a hydrophyte, your stomata will in fact be on the upper surface. But for mesophytes, your stomata are on the lower surface. Okay, so gaseous exchange is going to take place, and so you need a large intercellular air space for those gases to move in and out of. Okay. You might find these epidermal hairs. Those are to prevent herbivores and insects and other things from getting in. Okay, so let's now look at this plan diagram. So there is our cuticle across the top. Underneath that is a layer of epidermis. Then we've got our two regions. Okay, there's our first one, our palisade mesophyll. Here is our large one underneath it, our spongy mesophyll. Both of these are chlorenchyma cells or specialized parenchyma cells. And we have our vascular bundles. And notice that the vascular bundle sits directly across the two, okay, across the two layers of mesophyll. And the line that divides those two is actually your cambium, your vascular cambium. So the top section is going to be xylem, the bottom section is going to be phloem. Then we have our epidermis along the bottom, and the epidermis along the bottom has these specialized guard cells. Okay, I said to you those are the only cells that we actually draw as actual cells. And then in between those guard cells is a space, which is your stoma, or plural stomata. Pause the diagram now and make sure that you've got a copy of this plan diagram of the dorsiventral leaf in your books. If we look now at stems, obviously there are dicot and monocot stems. You'll be pleased to know that we're only going to expect you to know the dicot stem. How do you know the difference between a monocot and a dicot stem? It's where the vascular bundles are. So when we look at a dicot stem, you'll see that your vascular bundles are arranged in a circle around the edge of the stem. You have your xylem on the inside and you have your phloem on the outside of that. And then in fact, between those two, dividing those two, you would then have your ring of vascular cambium that runs through every vascular bundle. This is a cross-section of a micrograph showing a vascular bundle or showing a dicotyledonous stem, and you can see the vascular bundles. So there and there and there. Each of those is a vascular bundle. And you can identify the vascular bundle. These ones are obviously your parenchyma cells, your isodiametric cells, cells that are as long as they are wide. These cells all around the edge here, those are going to be your collenchyma cells. You can tell that because they're much darker in color, which means that the cell walls have stained. If you remember, your collenchyma cells have unevenly thickened cell walls. Then you can see here these very big cells. Okay, those are your xylem vessels. Then you've got a kind of layer through the middle. That is your cambium, which runs all the way around. Then you've got your parenchyma cells sitting just above that, and then a cap on the top of each. Again, these much darker cells, those are your sclerenchyma cells. Okay, and your sclerenchyma, if you remember, are evenly thickened around the entire thing. Right, when we draw our dicot stem cross-section as a plan diagram, again you have your epidermis on the outside, underneath that a layer of collenchyma, and then obviously all this packing tissue parenchyma. Now your collenchyma and parenchyma out here is what we call the cortex. Then in the middle you have a large circle of, again, parenchyma, which is known as your pith, and then all your vascular bundles that sit around the edge. When we look at the vascular bundle, you've got your xylem and your phloem. In between that is cambium, and then you might have a bundle sheath around it, and you might have a sclerenchyma cap on top as well. So here is your epidermis. Let's make that a different color so you can see it. Okay, there's your epidermis and your cuticle. Here, this is your collenchyma there, and this is your parenchyma there. There's your sclerenchyma cap. There is your phloem, there's your cambium, there's your xylem, and then obviously this is all pith parenchyma. Okay, again, I would pause the video at this point and make sure that you've got a copy of this plan diagram in your book for the stem. As a little exercise, again, pause the video at this point and use this diagram and draw your own plan diagram of this particular cross-section of a dicot stem. When you've finished your drawing, you can then check the memo on the next slide. So this is what your memo would look like. Notice there are no cells. We may have drawn one or two individual cells just to give some kind of perspective, but the vast majority of it is just a plan indicating where the different tissues are found. Right, let's look now at a vascular bundle. Again, pause the video, use this image and draw a plan diagram of where the different tissues in your vascular bundle are found. When you finish doing your plan diagram, you can then check the memo on the next slide. So again, we've drawn a couple of cells here and there just to give an indication of the size and the shape of the cell. But the major feature of the diagram is that it's a plan. There's an outline showing you where the different tissue types are found. Okay, let's move on to talk about roots. Again, dicot and monocot roots, we're only going to focus in on the dicot root, and again, the way that you would tell the difference is where the vascular bundles are positioned. So there are two new terms that you have to learn when we talk about root structure, and that's your endodermis and your pericycle. And they become very important when we talk about water movement in the plant, which is in our next video. So the endodermis, it's the last layer of the cortex. It's the boundary between the cortex and what's called the stele of the root. Notice the spelling of stele, S-T-E-L-E, not S-T-E-A-L. It produces a waxy layer, and again, water can't move through that waxy layer. We call that waxy layer the Casparian strip. And what that does is it directs the water into the vascular bundles. Again, I'm going to go through that in the next video when we look at water movement. The pericycle is the layer immediately inside your endodermis. It's part of your meristematic tissue. It's the first layer of cells in the stele, and when the root develops, that is where your lateral roots will come from. All right, so here's a cross-section of a ranunculus root. A ranunculus is a dicot, and you can see quite nicely here in the middle. Okay, you can see your endodermis there, and you can see your massive xylem vessels making a kind of cross or a star in the middle of the root. That is the dead giveaway that you're looking at a dicot stem, is that the vascular bundle of the root forms this kind of cross or star. So again, let's look at a plan diagram. But first, let's just focus in on the structure of this vascular bundle itself. You can see your vessel elements in the middle with their lovely thickened walls, secondary thickening of those walls. On the outside of the stele, you can see the endodermis, also lovely thickened walls that forms the Casparian strip. Immediately under that, you can see a layer of nice large cells. Those are your pericycle, which, as I said to you, that's where your lateral roots are going to start forming. Now, if you look in between the xylem, so the xylem forms this kind of cross through the middle, and then in between the arms of the cross, you've got these collections of cells, and when you look at them more closely, you will realize that they are your phloem cells. You'll be able to see some of the sieve tube elements, you'll be able to see some of the companion cells that are very small, and you'll be able to see the phloem tracheids. On the outside of your stele, you've obviously got lots of parenchyma cells, the isodiametric, very large cells. You don't need to worry about amyloplasts. Amyloplasts are storage parenchyma. It's just a specialized parenchyma, and you can see all of these starch granules, starch grains, being stored in those parenchyma cells. All right, so here is the plan diagram of the dicot stem. From the outside, you've got an epidermis, followed by the cortex, then your endodermis. The stele is made up of the pericycle, the xylem, and the phloem. Again, make sure that you pause the diagram, pause the video, and get a copy of this picture in your book. You can be asked to draw the plan diagrams for the leaf, the root, and the stem for a dicot plant. And as I say, plan diagrams, not actual diagrams. So you need to know the position and the labels of each layer, of each tissue in each of these organs.